This lecture is the first part of renal physiology consideration. It gives the general characteristics of kidney structure and function. In the second part, the regulation mechanisms for kidney function are going to be considered in detail. This first part of the lecture describes regulation mechanisms as well, but shortly. Excretion. This general function is performed by a number of organs in the body. This is the elimination from the body of waste materials that are ingested or produced by metabolism. The main end products of metabolism are carbon dioxide, CO2, water, H2O, H+ ions, hydrogen ions, and a number of nitrogen-containing substances which are products of the protein metabolism. These substances include urea, creatinine, and uric acid. Also end products of hemoglobin breakdown, mainly bilirubin, and metabolites of various hormones. A number of organs is involved into excretory function. But number one is kidney, major excretory organ of the body. Also, excretory function is performed by skin, mainly by glands, sweat glands, and as well sebaceous glands a little bit. Then lungs, which we studied already, that excrete most of CO2 produced in the body, as well as partly the water vapor. And gastrointestinal tract. So all these organs are involved into excretory function. Excretory functions of skin, lungs, and gastrointestinal tract should be mentioned briefly, and then we can switch exactly to the kidney physiology. As for the skin, mostly it refers to sweat glands, which excrete certain amount of water together with sweat, and also sodium and chlorine. Also, very little amounts of nitrogen-containing compounds can be performed by skin, such as urea, uric acid, and so on, but very little amount. Lungs excrete almost all CO2 and certain amount of water vapor, about half of a liter daily, and also when necessary, they can excrete some volatile substances that can produce vapor in the air, such as alcohol, ether, and some others. Glands of the gastrointestinal tract are able to excrete some medicines, drugs, morphine, quinine, salicylates. Also, it can excrete heavy metals, salts, and other foreign substances, but naturally, on a daily basis, liver excretes bilirubin and also the excess of cholesterol with bile. Main excretory function of kidney includes excretion of nitrogen-containing metabolic end products. As it was already mentioned, that skin can excrete certain amount of these substances, but negligible amount, and kidney makes the most of excretion of these substances. Urea, uric acid, and creatinine, and also some foreign substances, and plus ions and water. Main way of excretion of ions and water is through kidney. By excreting or conserving the correct amounts of water and dissolved substances, which are called solutes, the kidneys regulate the composition and volume of body fluids. Regulatory homeostatic functions of kidney. Along with the main excretory function, the kidney performs numerous other functions, regulatory functions. And there is one big group of homeostatic function. Homeostatic function is not the only one. It is composed by a number of separate functions. Let's list these functions. General regulation of water and electrolyte balances. It's a very general function, but mostly it refers to regulation of such an important constant of the body, which is osmotic pressure, or simply osmolality, because ratio of water and electrolytes is exactly measured by osmotic pressure. Regulation of body fluid volumes. It includes the extracellular fluid volume And as a part of this extracellular fluid, the blood volume, important function of kidney. Regulation of arterial blood pressure. It's based on the previous regulation of fluid volume, because mostly pressure depends on one of the most important factors for blood pressure is the blood volume. Regulation of acid-base balance. Kidney are able to excrete excessive amount of H plus ions or less excrete, and H2O3 minus ions can be excreted predominantly. And participation in constant level of blood glucose, regulation of blood glucose by the way of gluconeogenesis. This is production of glucose from amino acids predominantly or other non-carbohydrate sources. And kidney can do it in rather big amount, which is comparable to liver ability for gluconeogenesis. Then there is a big group of endocrine functions of kidney. And it is performed by releasing into circulation the following substances. Renin, the enzyme that causes angiotensin 1 formation from angiotensinogen. Angiotensin is blood plasma protein which circulates constantly in blood, but when renin appears in blood, it splits angiotensin 1 from this big protein, and it starts the development of all processes that we refer to renin angiotensin aldosterone system, RAAS. Erythropoietin is secreted by kidney under hypoxia. Hypoxia is the main stimulus for erythropoietin secretion, and in turn this substance stimulates production of red blood cells in the bone marrow. Then, formation of active form of vitamin D, calcitriol, or 1.25 dihydroxy vitamin D3. This last hydroxylation in the first position occurs exactly in kidney. So the active vitamin is formed in kidney. And as you remember, this active form of vitamin is called hormone calcitriol, which is essential for normal calcium deposition in bone, and also is the main factor that absorbs calcium in gastrointestinal tract. The structure of the kidney. Generally, kidney can be divided into renal cortex and renal medulla. Emptying of tubules goes into renal pelvis and then finally into ureter that goes to bladder, urinary bladder. If we enlarge one portion of renal cortex and medulla, we can observe numerous functional units of the kidney, which are nephrons. They are shown in yellow here, and now in red circles you can see the glomeruli, which are numerous and they are main for process of filtration in kidney. The nephron, basic functional unit of the kidney, and it consists of many parts. And first, nephron consists of the Bowman's capsule, which surrounds each glomerulus. Then, tubule starts out for capsule of Bowman, many tubules. And first tubule is the proximal convoluted tubule, which is located exactly in the cortical part of kidney. Then, tubule goes downward to the medulla and forms the loop of Henle, which consists in the 3 portions. Basically, of course, first we can divide into 2 portions, descending and ascending, but the ascending part is divided again. Descending part, then the thin ascending number 4 and the thick ascending segment of the ascending limb of the loop of Henle. Then the loop of Henle returns tubule back to cortical part of the kidney, and there is a distal convoluted tubule. After this, tubule again goes downward to the pelvis, where few nephrons can be emptied into one and the same collecting duct. Therefore, formally, collecting duct is not a part of individual nephron, but functionally, of course, collecting ducts work together with the whole tubule system. Many nephrons empty their distal tubules into one and the same collecting duct, which carries the final urine to the renal pelvis and ureter. Blood supply to a nephron. Each nephron has its own blood supply, which is composed of 2 arterioles and a 2 capillary system. It's an unusual case. We have just about 3 such cases in the body with 2 capillary systems, and it's the only one case when there are 2 arterioles. The first arteriole is broken into numerous capillaries of the glomeruli, but then its going out vessel is again arterial. So the blood is still rich in oxygen and is supplied in the second outgoing arterial, efferent one, and then again it is broken into many capillaries which are peritubular. Let's see. Bowman's capsule surrounds each glomerulus. This is space for filtration. The bringing or efferent arteriole enters the glomerulus and forms really glomerulus of capillaries, which participate in filtration process. Then they join again into arteriole. Now it's outgoing or efferent arteriole. And then this efferent arteriole forms the network of peritubular capillaries. And here, peritubular capillaries finally become converted and come to venules. So blood gives oxygen and it becomes venous. Exactly in peritubular capillaries, the processes of taking back of many filtered substances occur, and this process is referred to as reabsorption. There are subtypes of nephrons, and we can divide them into cortical and juxtamedullary. The cortex can be indicated here and medulla. The cortical nephrons are shorter and they make the majority, about 80% of nephrons. And juxtamedullary make about 20%. Cortical nephrons have shorter loops of Henle and peritubular capillaries like a net. They are convoluted enough, these capillaries. While the juxtamedullary nephrons have much longer loops of Henle that go deeply into the medulla, and they are surrounded by peritubular capillaries which are called vasa recta. They are much more straight and go deeply downward to the medulla and then come back to venules in the cortex of nephron. And the glomeruli of the juxtamedullary nephrons are located on the border between cortex and medulla. Basic processes that occur in the kidney. There are 3 such basic processes underlying the urine formation. First, it's glomerular filtration. A lot of blood plasma is filtered through kidney filter into the space of the Bowman's capsules, and then it's supplied into tubules. And in the tubules, the process goes for tubular reabsorption. But also there's a sad process. It's a tubular secretion, and it's necessary not to mix it with any other secretion such as endocrine secretion. It's exactly excretory secretion, tubular secretion, in the direction of this process is opposite to reabsorption, while reabsorption is the movement of solutes, dissolved substances, and water from the lumen of a tubule into the interstitium and finally to blood. Reabsorption goes back into blood. And the secretion is opposite transport of substances from blood to the interstitial fluid and then to the lumen of a tubule. So the secreted substances are going to be excreted and removed out of the body. These 3 basic processes of urine formation are shown in the picture now. Simplified picture where you can observe the Bowman's capsule, where filtration occurs from blood capillaries in the glomerulus into the Bowman capsule space. Then the piece of tubule is shown, where process of reabsorption is shown by arrows out of lumen. Actually, the arrows should be directed into blood vessels surrounding peritubular capillaries. And secretion shown by arrows oppositely directed into the lumen, the lumen from blood. This is tubular secretion, kidney secretion, which is not endocrine secretion at all. Finally, what remains in the tubules is excreted. So, as a result, we can express excretion as the product of filtration minus reabsorption and plus secretion. Substances which are filtered and not fully reabsorbed, and plus substances which are secreted can be excreted out of the body. High renal blood flow is the necessary condition for kidney function. Kidney itself does not need so much oxygen, but it's the function of kidney to filter blood many times daily for its filtration and then to produce reabsorption and formation of the final urine. There is about 25% of cardiac output, kidney blood supply. From at least 20 in some sources they give up to 25%. So we can calculate if we take 25% out of regular average 5 liters per minute cardiac output, it makes 1.25, or at least, if 20% is taken, at least 1 liter each minute. This is the amount of blood that passes through the kidney each minute, about 1.25 of a liter. And this renal blood flow is rather constant over the wide range of changes of the general arterial blood pressure, systemic blood pressure. And this allows kidney to have constancy of final excretion processes. Renal artery is subdivided into numerous segmental arteries, then interlobar, then arcuate, and finally interlobular arteries. And then they form the arterioles that supply blood into each glomerulus. Glomerulus has such a structure that has great surface area for filtration. Note that the glomerulus itself has the total surface area much greater than the capsule of Bowman. It doesn't, in spite of the fact that it's located within the capsule, its total surface area is the surface area of all the loops of capillaries that compactly make the glomerulus. And each individual capillary loop each capillary is covered by filtration, forms the filtration membrane. It's covered by epithelial cells. And epithelial cells make the third layer of filtration membrane. And these epithelial cells are called podocytes, which can be translated word for word as cells with legs. And podocytes inside the glomerulus that surround each glomerular capillary, many of such cells form the filtration membrane, the third layer, and this filtration membrane is represented by pores between processes. The body of podocytes produces primary processes, which are large enough, and from this primary process, the finger-like smaller processes or food processes come to all the sites, and they are interdigitate with other processes of other cells, so that very narrow but long slit pores are formed. Here you see the food processes, and slit pores can be observed between meeting each other food processes. They are wide enough, and the probability of these slit pores is very high. So many substances can pass through these slit pores. Here you saw the schematic presentation of the filtration membrane of glomerular capillaries, and here is the actual view of the scanning electron micrograph of glomerular capillary. And you can see here podocytes cells body. You can see the primary processes larger and small branching food processes of pedicells shown here. And slit pores are visible between these last processes. Let's consider the full structure of the glomerular filtration membrane. We take a little piece of the glomerular capillary and enlarge it, so that we see schematic representation of all the three layers. So filtration goes from blood into the Bowman's capsule. And first layer is endothelial cells, made of endothelial cells of a capillary. Between cells, the pores are very large, and they are called fenestra. Fenestration is important and provides great openings through which many substances can pass. Then, next layer is the basement membrane. Basement membrane also has large enough pores, but they are smallest among all the pores, all the layers of the filtration membrane, smallest pores. And then finally epithelial cells, which are podocytes. And when we make cross-section, we may not see full cells, and just pieces of podocytes sometimes occur in the cross-section. But between the podocytes' legs, between their processes, slit pores are widest among all the layers of filtration membrane. So here again you see the electron micrograph of the filtration barrier. Here you can observe how, from at the bottom you can observe the vessel with red blood cells portion appearing just in the bottom of the picture, and the layer of endothelial cells can be seen, and between endothelial cells there are pores through which filtration of water and dissolved substances may occur. So all three layers can be seen here: endothelial cells, and through pores between them many substances are able to pass. Then basement membrane is shown with smaller pores but also possible for many, permeable for many substances. And finally slit pores between food processes of podocytes can be seen, through which the substances can be filtered from blood, from capillaries into Bowman's capsule. So the glomerular filtration follows the same laws as filtration from other capillaries, as we consider in microcirculation. And we consider the Starling equation. This equation contains coefficient of filtration and number of pressures. The four pressures, actually, two types of pressures: hydrostatic and colloidosmotic or simply oncotic pressures participate. But usually we have the four pressures because pair of each pressure is present inside the capillaries and outside the capillary. And here we have hydrostatic capillary pressure, filtration coefficient of course first, hydrostatic capillary pressure, and hydrostatic pressure in the filtered fluid in the capsule. Then we have these two pressures are opposite to each other. Then we have the oncotic or colloidosmotic blood pressure, which is directed inside the capillary, and there is no oncotic pressure in the filtered fluid because filtrate does not contain proteins. No protein, pressure is zero. So as a result, the Starling equation is converted in this case into shorter equation without last pressure. So just the three pressures participate in filtration processes in the kidney, because oncotic pressure is not present in the filtered fluid. So forces that cause filtration. The values for humans are estimated only approximately based on the results on experiments in animals. And approximately values are as follows. Force that favors filtration, actually the only one. This is glomerular hydrostatic pressure. Hydrostatic pressure inside the capillaries. Usually capillaries have pressure about 30 to 35 millimeters, and this pressure is only for arterial end of a capillary. And in the venous end it was 15 to 20 or sometimes even less. Here pressure is higher than regular pressure in average capillaries in the body. This pressure is about 60 millimeters mercury. There are few explanations of this. One explanation says that the renal artery comes from the abdominal aorta straightly and is short enough, and pressure does not fall lower as for many other capillaries in the body. And there is one more explanation, that the outgoing artery from each glomerulus is less in diameter than bringing efferent arteriole. Therefore, there is higher pressure inside the glomerulus. And this pressure is considered to be about 60 millimeters hydrogen. And Bowman's capsule oncotic pressure, which theoretically, if it's present, could contribute also to the filtration, but this pressure is 0. Therefore, among 3 pressures which participate in formation of the final net filtering pressure, there is only one that contributes to filtration, favors filtration. Opposing filtration, there are 2 pressures. Glomerular capillary oncotic pressure created by blood plasma proteins. It's about 32. It's slightly higher than usual oncotic pressure because of loss of fluid, and proteins become slightly more concentrated in the glomerular capillaries. So that pressure is slightly higher. Usually it's 25-30 millimeters, and now it's 32. Bowman's capsule hydrostatic pressure is about 18. So these values, of course, are not stably exactly on these values, but plus minus they are variable around these average values. And therefore we can calculate the final pressure which contributes to filtration. Net filtration pressure. If hydrostatic blood pressure in the capillaries of glomerulus is 60, then opposing oncotic pressure of blood is 32, and pressure in the Bowman's capsule, which is also opposes, is 18 millimeters. Therefore, total net filtration pressure is 60 minus 50, or simply 10 millimeters mercury. 10 millimeters. This pressure is enough to produce filtration as we have it normally in the kidney every day. 10 millimeters. And if any individual pressure among the listed 3 components can be more or less changed, this net filtration pressure, resulting pressure, usually is maintained on approximately the same level. Otherwise, the processes will become changed, and therefore there are numerous mechanisms that maintain this constancy of the filtration pressure, and finally constancy of the filtered portion of blood each minute, glomerular filtration rate. Factors affecting filtration pressure. Actually, all the 3 components can affect this pressure, but which changes may produce changes of these factors? First, capillary hydrostatic pressure can be increased or decreased. For example, it can be increased by the dilation of the afferent arteriole. If this arteriole is dilated, it allows to flow more blood into the glomerulus, and thus glomerular volume and pressure increases. Also, it can be increased by the constriction of the efferent outgoing arteriole, and if it's constricted, outflow is obstructed, it becomes less, and again filling of each glomerulus becomes higher and pressure increases. Then, opposing oncotic blood pressure depends on the blood protein concentration, and in case of great changes of the oncotic pressure, we can expect changes of the net filtration pressure. Also, opposing Bowman's capsule hydrostatic pressure can be increased by constriction or blockage of ureters. First, of course, pressure increases in the renal pelvis, then in collecting ducts, but gradually it may reach the Bowman's capsule, where it becomes higher and opposes the filtration, so that the filtration decreases as a result of higher opposing pressure in the Bowman's capsule. Glomerular filtration rate. Glomerular filtration rate is approximately about 125 milliliters per minute. It means that exactly this amount of blood plasma enters the Bowman's capsule. And if it's recalculated, if these minutes recalculated into hours and finally into day, it makes 180 liters of filtrate per day. 180 liters. But generally these values are for men. For women we have about 110, 115 milliliters per minute, and correspondingly somehow less liters daily. But for memorizing it's easy, 180 liters of the primary urine, which is the filtrate, primary urine. And of course it's clear that such a great amount of fluid we cannot lose, and we must reabsorb most of this volume. Actually 99% on average of all this fluid we reabsorb daily, and about 1 liter approximately, 1% approximately from this volume plus minus we excrete finally as the final urine. So there is a great task for reabsorption of mainly almost all this fluid. The fraction of the renal plasma flow that is filtered, or filtration fraction, is about 0.2. It means, for example, when 1 liter of blood plasma passes through the glomeruli, about 200 liters are filtered into Bowman's capsule, and actually 20% of each volume that passes through the kidney. About 20% filtered from capillaries into the Bowman's capsules of totally all glomeruli in the kidney. Let's observe the filterability of substances by glomerular capillaries. As you remember, usually capillaries allow easy passage of water and ions, but substances a little bit higher, even glucose, which normally should easily go out of capillaries, already has less filterability as compared to water. It will be 0.8 approximately for glucose, and progressively less and less for each higher and higher substance. Here you can observe that substances such as water and sodium pass the same as water itself. Then glucose, which is large enough, 180 molecular weight, also passes the same as water, and even inulin, which has molecular weight 5,500, filters exactly the same amount that water molecules. So all low molecular substances, from really very low to higher molecular weight substances, all equally easily pass the glomerular filter. And filtrability is the same as for water. Then proteins, myoglobin and albumins, are given for comparison. Myoglobin is relatively small protein with molecular weight about 17,000, and you see it's filtered not perfectly as water but rather high, 0.75. So out of 4 molecules, each 3 molecules will be easily filtered. But fortunately our blood does not contain myoglobin. Myoglobin is located inside the muscle cells, therefore it should not appear in blood and cannot be filtered normally. Albumin, this is normal blood plasma protein with molecular weight about 69,000. And you see albumin filterability is 0.005, which means 1 molecule out of 200 can be filtered. So it's negligible amount. So generally blood plasma proteins cannot be filtered by kidney filter. With smallest blood plasma protein, albumins, which can be filtered in negligible amount, but larger proteins such as globulins or fibrinogen entirely cannot pass through kidney filter. So proteins filterability for blood proteins is negligible or absent entirely. So the filtrate composition is the same as of blood plasma except for proteins, because even smallest blood plasma protein, albumin, is filtered 200 times less than glucose, sodium or water. Filtrate reabsorption to the blood of peritubular capillaries. Look at the peritubular capillaries. Now these capillaries become converted gradually to venous end and venules. So here we have much greater pressures. For capillaries in the glomerulus, blood pressure in these capillaries is 60 millimeters mercury, and it's the same when blood flows out. So pressure remains unchanged, but when blood enters the peritubular capillaries, pressure becomes much less. In the arterial end of these capillaries is about 18 millimeters, and in the venous end it's 8 millimeters mercury only. So this low pressure contributes to easy process of reabsorption. The substances can be taken to these capillaries easily because of their low pressure. But anyway, for many substances it requires active transport. Filtration occurs in the glomeruli, while reabsorption occurs in the peritubular capillaries. Also, there is process of secretion, not shown in this picture, but you may remember that tubules, not all of them, but some of them, can secrete substances into the lumen. But first we're going to consider tubular reabsorption. It's much more complex for number of many processes that occur differently in different tubules. So that we need to recall again which parts of the nephron tubules we have. First is the proximal tubule, which is convoluted so that it can be long enough, but takes not much space, occupies not much space. And these proximal tubules are located in the cortical part of the kidney structure. Then, loop of Henle. First part is the descending limb. Then, second part is the thin portion of the ascending limb. And final portion of the loop of Henle, thick ascending limb, which actually contains mechanisms that drive the whole processes inside the loop of Henle. Then there is next part, distal tubule, also convoluted and also as proximal, located in the cortical portion of kidney structure. And collecting ducts finish the processes of reabsorption, going downward to the renal pillars. And first we're going to consider the reabsorption processes in the proximal tubule. Here in one and the same picture, in the same proximal tubule cell, many mechanisms of reabsorption are summarized. To the left side from this cell we have the lumen of the tubule. To the right side, this is the basolateral membrane which faces the blood vessel, the peritubular capillaries. And the main structure which uses energy constantly here is the sodium-potassium pump, regular sodium-potassium ATPase, that transports both ions against their concentration gradients. Sodium goes into interstitium and finally joins the extracellular space and become evenly distributed between blood and extracellular fluid or interstitial fluid. Concentration cannot be different, so it immediately appears to be reabsorbed after passing through the membrane, basolateral membrane of the proximal tubule cell, while potassium goes into the cell against gradient for potassium. And due to this, sodium concentration is maintained low in the cell constantly. For sodium, transport is considered to be primary active. Doesn't matter that sodium can enter from the lumen into the cell using protein carriers, can enter passively, but totally passing to the proximal tubule cell requires energy for pump operation. Therefore, sodium is transported by primary active transport. Exactly on a basolateral membrane, ATP is spent for transportation of sodium. But due to low concentration of sodium, protein carriers easily transport sodium into the cell, and these different types of carriers transport together with sodium, along with sodium, the other molecules. Of course, this is a primary active transport, and now we observe also sodium is transported partially by exchanger. Sodium enters the cell due to its low concentration in the cell in exchange with the other positively charged ion, proton of hydrogen, H+. And this is counter transport because sodium is transported into the cell and hydrogen out of the cell. This is one of ways of entry of sodium into the cell. But then sodium carrier proteins transport along such substances as glucose, amino acids, and some ions, some salts. So this is exchanger, and now we should observe the passive transportation of sodium into the cell by protein carriers with glucose. Then other transporters transport amino acids Other transporters can transport such substances as phosphate, lactate, or citrate. All these way of transport for these substances, glucose, amino acids, phosphate, lactate, and citrate, this is the secondary active sodium-dependent transport. Then the same substance, glucose, amino acids, and so on, transported to blood by their own protein carriers. For glucose separately, for amino acids, for phosphate, lactate, or citrate. And also HCO3- ion are transported by protein carriers. So among these listed substances, the only one substance is transported by primary active transport. This is sodium. While glucose, amino acids, phosphate, lactate use secondary active sodium-dependent co-transport. Co-transport because the direction of transition of substances, transportation is the same into the cell. So there are numerous protein carriers used in both luminal and basolateral membranes of the cell, both sides. Reabsorption of proteins. As you remember, one of each 200 molecules of albumin approximately can pass into the glomerulus capsule and go into tubules. And these protein molecules are reabsorbed exactly in the proximal tubule by endocytosis. Here you see the cross-section, which is in horizontal plane now, so that you can observe the cross-section of the lumen, tubular fluid in the lumen. This is primary urine. And you can see the tubular cells that take these protein molecules by formation of the coated pits. Then endosomes are formed, or early and late. They join the lysosome, and finally, inside the lysosomes, by their proteases, the proteins, albumins, are digested into amino acids, and these amino acids are released into interstitial fluid, and finally they go to blood. As protein molecules are too large, there is no other way of transportation these molecules through the membranes of cells of the kidney tubules. So only endocytosis, or it can be said pinocytosis, is used for proteins reabsorption. And actually all the little amount of protein molecules that can pass into filtrate, all these molecules normally are reabsorbed in the proximal tubule. Reabsorption of filtrate in the proximal tubule occurs here, it's shown in red, the proximal tubule, and as for sodium and water as well, is 67% approximately of the whole filtered fluid. 67, or simply 2/3. 2/3 of all filtrate is reabsorbed exactly in the proximal tubule. The same amount of sodium in percent and of water percentage, the same because the proximal tubule cells membranes are fully permeable for water. Therefore, each ion that goes out of tubule lumen is also followed by water. It's surrounded by water molecules which, due to osmotic reasons, leave the tubular capillary, tubular fluid into capillary, leave proportionally to sodium reabsorption. So due to osmotic forces, reabsorption of sodium is followed by water reabsorption proportionally. Therefore, the osmotic pressure of fluid that passes through the proximal tubule remains unchanged. That this tubular fluid loses sodium and other ions, but in proportion it loses water. Therefore, the osmotic pressure remains unchanged. The transport maximum for reabsorbed substances. It's important. The reabsorption of most of these organic substances is limited by the number of carrier proteins, as reabsorption occurs using these carrier proteins, and these carrier proteins have certain number, and therefore they can function with limited range. The concentration of substance, if it becomes higher and higher, it requires participation of more and more carrier proteins for their transfer through membrane. and these carrier protein numbers finally can reach them all involved, and further reabsorption cannot be performed. So transport maximum is reached. It's named the transport maximum. Concentration of a substance at which reabsorption becomes already not complete, so that excretion of the substance begins, it remains in the final urine excreted. So it's referred to as the threshold of excretion. Mostly it can be, of course, be observed on many organic substances, but most often it's considered on the example of glucose. Glucose excretion threshold. And glucose, as you understand, is important for the body substance, and even when its level is higher than normal in blood to a certain degree, we still try to preserve it in the body. So it means that we have protein carriers in such amount that it's more than enough for transportation of glucose when it's normal. And they can become enough even with elevation of glucose level higher and higher progressively. But finally, participation of all carrier proteins can be reached, and then any excessive glucose over this level will remain unreabsorbed, because all carrier proteins are already busy with reabsorption, and any more molecules will not be fully reabsorbed. Here you see filtration, red line, which goes straightly, and the high is glucose concentration, which goes on horizontal axis. When it becomes higher, it becomes more filtered, because glucose filterability is 1, the same as water. And so it simply proportionally becomes more and more filtered. But reabsorption, shown in blue line, goes parallel to filtration until certain degree, and then transport maximum is achieved, and there are no more additional free protein carriers. Therefore, the glucose reabsorption reaches the maximal, which cannot be exceeded. And therefore, from this certain moment, when reabsorption maximum is achieved and filtration still goes upward because of higher and higher glucose concentration, we can see that threshold is reached and the excretion begins. Moment when excretion begins, it's the moment of threshold of glucose. And here on the axis you can see in milligram per 100 milliliters, but mostly we use for glucose millimole per liter. And glucose excretion threshold is about 10 millimole per liter. Of course, with certain individual differences, but generally we consider that it's about 10 millimole per liter. And you see this threshold is higher than normal range of glucose, at least for fasting state. Much higher than normal level of blood glucose, which is 3.3 up to 5.5 millimole per liter for the arterial blood. So you see, even when glucose becomes 6 millimole per liter, 7, 8, 9, still glucose is fully reabsorbed. It shows that carrier proteins are enough. Additional carrier proteins can be included into functioning, and glucose anyway is reabsorbed. But when level reaches 10 millimole per liter, so there appears not reabsorbed glucose, and taking into account that only proximal tubules have the carrier proteins for glucose. Any glucose molecules that escaped into loop of Henle further don't have any chance to be reabsorbed, and therefore glucose appears in the final urine. Thus, in the proximal tubule, for many organic substances, reabsorption is fully completed. Glucose, amino acids, proteins, the negligible amount of proteins that may enter the filtrate. Then phosphate, lactate, and citrate. For all these substances, reabsorption is completed exactly in the proximal tubule. So the filtered fluid that flows further into the loop of Henle normally does not contain any of these substances, because 100% of them is reabsorbed in the proximal tubule. And as for sodium and water, reabsorption is 2/3. Therefore, for all remaining parts of the nephron tubules, we need to reabsorb the 1/3 remaining. If you recall that total volume is 180 liters daily, it means that 1/3, 60 liters, are to be reabsorbed in the next parts of the tubules, in next tubules of the nephron. And let's go to next part of the nephron, loop of Henle. Thin descending limb, first portion of the loop of Henle, is highly permeable for water, and water goes down the gradient of the osmotic pressure. It goes to the side where number of ions of osmotically active substances is greater, and higher osmotic pressure is around the tubule in the interstitium of medulla. Its hypertonicity of interstitium is maintained constantly, regardless of new and new portions of water that come from the descending limb of loop of Henle. Water goes and goes out of the descending limb. And ascending limb, both the thin and thick portions, are entirely not permeable for water. Here is shown by the dark blue line, and you see it extends even into the distal tubule part. Early distal tubule is not permeable for water as well. So you can understand that water reabsorption cannot occur here at all. But instead, there are active processes of ions transportation, and only thick portion of the ascending limb of loop of Henle reabsorbs sodium, potassium, and chlorine. That each molecular device that transports this ion transports in one cycle two positive ions, sodium and one potassium, and two negative ions, both chlorine ions. So 4 ions are transported per each cycle, and they create this hyperosmolarity or hypertonicity of the interstitium. Just if you consider it in order, first descending part, then ascending. And it looks like reason is later than consequence of water reabsorption. But in real kidney, all these processes occur simultaneously. The filter runs through the tubules constantly, and ascending limb constantly removes sodium, potassium, and chlorine out of filtered fluid, and these ions are located in the interstitium on their way to blood, and therefore interstitium constantly has high osmotic pressure, and this high osmotic pressure contributes to water reabsorption passively from descending limb of the loop of Henle and also from the collecting duct. Just in the collecting duct, the permeability for water can be variable. Here you see the mechanism of work of the thick ascending limb cells. There is a co-transportation of sodium, potassium, and 2 chlorine ions. And this mechanism molecular device is located on the luminal side of the membrane. Sodium, chlorine, and 2 chlorines and potassium. And itself, this transporter does not use energy. You see there is no ATP written in the middle. But it's all based on sodium entry to the cell down the gradient. And this gradient will quickly disappear if sodium constantly goes and goes into cell. But sodium is constantly pumped out of the cell by regular sodium-potassium ATPase, or sodium-potassium pump. And this is the main force where we spend energy, and this energy finally maintains constancy of the co-transport of sodium, potassium, and chlorine from the lumen through the cell and outside. Also, you should know for future study of pharmacology that exactly this sodium, chlorine, potassium transporter can be blocked by such medicine like furosemide, due to its operation exactly within the loop, within actually not simply loop but thick ascending portion of the loop of Henle. It's called the loop diuretic. And it's not the only one, but it's a main representative of the group of substances that are used as increasing diuresis or urine formation and release from the body, furosemide. And it acts very actively and very quickly because it blocks the main mechanism of reabsorption in the loop of Henle, and also it influences the collecting duct. Now we need to consider in details this system of operation of loop of Henle firstly, and also it operates together with the collecting duct, not shown at the picture. And this is a complex mechanism of related processes, which is referred to as countercurrent multiplier system. First, why it's countercurrent? It's easy to understand because the filtered fluid flows first downward to the renal pelvis through descending limb, and then it goes upward to the cortex through ascending limb. This is countercurrent. And while the fluid flows through this tubule, through the whole loop of Henle, the processes increase each other. The process that occurs in the ascending thick portion of ascending limb, this is a driving process for the whole multiplier system. And let's observe first from the beginning. First, fluid that enters the proximal tubule is blood plasma, just part of blood plasma, and its osmotic pressure is exactly the same of blood plasma. If you remember, it's 290 plus minus 10, but here we expect such great changes of the pressure, so we can simply round it to 300 millismol per liter. And as you remember, proximal tubule reabsorbs ions and water proportionally. Therefore, the osmotic pressure remains unchanged while fluid flows through the proximal convoluted tubule. And therefore, in the entry to the loop of Henle, due to parallel reabsorption of both sodium and water, osmotic pressure remains the same, about 300. But then, while fluid flows down to the through descending limb, water is reabsorbed and ions are not reabsorbed, only water. And water is reabsorbed and taken to capillaries, to the whole extracellular fluid. And due to this reabsorption, the osmotic pressure becomes higher and higher because the same substances are dissolved in smaller and smaller volume. Due to loss of water, the osmotic pressure gradually increases and it becomes as higher and higher progressively with going down and down. And the deeper goes the loop of Henle into interstitial of medullary portion of kidney, the higher becomes osmotic pressure. So longer loops provide highest pressure in the tip of the loop, which is the most downward portion. You see 600, 800, 1000 millismol per liter, and we can reach even 1400 millismol per liter in the tip of the loop of Henle, which is the most lowest portion. So this portion of the loop of Henle produces great concentration, so it's called even concentrating limb because of only water reabsorption. But then, let's recall, from the moment when loop turns upward, the ascending portion is entirely not permeable for water. And instead of this, and instead of water reabsorption, here we have ions reabsorption. In the thick portion, ions are reabsorbed, such as sodium, chlorine, and also potassium, not shown here. And exactly now you can see explanation why the osmotic pressure is high in the interstitium. It's one of the main reasons of high osmotic pressure in the interstitium of medulla. And that's why water readily flows to all the side from the descending limb. This picture reminds you that all the limbs of the loop of Henle and generally all the loops of Henle are located in one and the same common interstitium. And if on even not all of these portions of loop and only ascending parts, thick ascending portions, produce constant reabsorption of sodium, potassium and chlorine, anyway, the totally interstitium becomes hypertonic. And therefore, in each descending limb, water readily goes out of the lumen. And then, when fluid passes up and up in the ascending limb, due to loss of ions and unchanged amount of water, concentration becomes progressively less and less. And when filtered fluid flows out of the loop of Henle, finally we have even hypotonic fluid, less than blood plasma, not 300 but just 100 milliosmol per liter. And that's why it's called multiplier system. You may understand now, if transport of sodium, chlorine and potassium out of the thick ascending limb of loop of Henle increases, the more ions are reabsorbed into interstitium, the more water becomes reabsorbed from the ascending limb, because water is reabsorbed proportionally to the difference of the osmotic pressures. So high hypertonicity of interstitium contributes to greater reabsorption of water. Therefore, system operates as not only countercurrent but as multiplier system. But again, driving force of all these processes is sodium, potassium and chlorine reabsorption, which occurs using molecular device which itself is only transporter, no use of energy, but this transporter may operate only when sodium-potassium pump is active and maintains low sodium concentration in the cells of the thick ascending portion of the loop of Henle. And due to this, water can be reabsorbed passively without energy spending from the descending limbs. So the key portion is thick ascending portion, which, as was said, can be blocked by the furosemide and other medicines of this type, loop diuretics. And this descending limb is considered concentrating limb, while the ascending limb is diluting limb, and as a result we can observe hypotonic fluid flows out of the loop of Henle. Reabsorption in early distal tubule. Early distal tubule is this portion that also is impermeable for water. Therefore, it also contributes to further dilution of fluid. And early distal tubule reabsorbs sodium and chlorine by cotransporter. Both ions are transported out of the lumen, sodium and chlorine. And so there is no loss of fluid, but there is loss of ions. So it again diluting action. It's still impermeable for water. So it continues to dilute filtered fluid. Further dilution. And the osmotic pressure initially was 100, already hypertonic, and now when fluid flows into the late distal tubule portion, which is already permeable for water, the osmotic pressure is even less, about 80 millismol per liter. Reabsorption processes in the late distal tubule and collecting ducts. This is the late distal tubule portion, and this is the collecting duct. The processes are very similar here, and what is necessary to mention first, that here processes of sodium and water reabsorption occur not proportionally, not necessarily proportionally, and regulated separately, regulated by hormones. And main hormones that are involved into this regulation are the following, which we already studied. Antidiuretic hormone, which contributes to water reabsorption, separately from sodium. It's important. And aldosterone is the second hormone, which contributes to the sodium reabsorption. To understand the functioning of these late distal tubules and collecting ducts, we need to know that there are two types of the cells in these parts of nephron: principal cells and intercalated cells, spaced between the principal cells. Now let's observe the functioning of these two types of cells. Principal cells, they reabsorb sodium and chlorine, also secrete potassium into the lumen. While and in case of ADH present, sodium and chlorine are reabsorbed, followed by water. Naturally, sodium should take water itself, but here water permeability can be regulated. If water permeability is present, water may follow sodium, but water permeability can be as low as simply 0. And in this case, reabsorption of sodium will not be accompanied by parallel reabsorption of water. That's how ADH regulates water reabsorption separately. Mechanism of water reabsorption itself is again due to osmotic reasons, but presence of the osmotic pressure difference can be followed by reabsorption of water and can be not followed if permeability for water is decreased or absent. That's how ADH may modulate amount of water reabsorbed. It can follow sodium, may not follow. Intercalated cells, they have different processes of reabsorption. They reabsorb potassium ions and HCO3 ions, while they secrete H+ ions into the lumen. This secretion can be high, can be less, and so it contributes to maintaining the normal acid-base balance in the body because we can excrete more acidic ions and less. Distal tubule and collecting duct processes of sodium reabsorption. Principal cells are controlled by aldosterone. Let's observe this mechanism. Aldosterone is the steroid hormone, therefore it's easily dissolved in membrane phospholipids. And so it doesn't use the outer membrane receptors, and it binds to intracellular receptors. When it enters the cell, it binds to the receptors in the blood, binds to the receptors located in the cell, not in the nucleus, but together with these receptors, it enters, cytosolic receptors, enter the nucleus finally, where it stimulates the synthesis of specific mRNAs. So it means that it simply stimulates finally synthesis of proteins. And these proteins, we know which protein is the number one for sodium reabsorption. It's simply sodium-potassium pump, sodium-potassium ATPase. This is a protein. And the more such proteins are produced and are inserted into basolateral membrane, the more sodium is pumped from the cell into interstitium, and it drives entry of more sodium into the cell from lumen. But for sodium entry to the cell, it's necessary to have opportunity to pass, but sodium charged particle cannot pass simply phospholipids of membrane. And it's necessary to have channels for sodium. And the other proteins additional to sodium-potassium ATPase, which is the key protein for sodium reabsorption, but also proteins that form sodium channels in the luminal membrane. These sodium channels allow sodium to enter the cell down the gradient, and then this sodium is pumped by the pump into interstitial fluid, where it becomes part of the extracellular fluid, and it becomes evenly distributed between between the interstitial fluid and blood plasma. So it becomes reabsorbed and remains in the body. It's lost from the lumen and goes into the blood. This is action on the principal cells, reabsorption of sodium, while potassium is lost outside into the lumen. Aldosterone action on intercalated cells. Intercalated cells reabsorb potassium and secrete hydrogen because the cells have hydrogen-potassium pump or hydrogen-potassium ATPase. These cells also are stimulated by aldosterone as well as principal cells, and therefore aldosterone also stimulates H+ ions secretion. Thus, aldosterone stimulates sodium reabsorption as well as water, but water reabsorption not necessarily follows the sodium reabsorption because it's necessary to have ADH enough. Water reabsorption is controlled by ADH. And also potassium and hydrogen ions are secreted. It's the opposite direction from blood into the lumen. So aldosterone influences the three ions at once, influencing principal and intercalated cells. Water reabsorption in distal tubules and collecting ducts is regulated by antidiuretic hormone. This hormone is the peptide, and so it's water soluble and not soluble in phospholipids of the membrane. Therefore, it uses the 7 TMS receptors, 7 transmembrane receptors located in the outer membrane of cells on the basolateral side, which faces the blood capillaries. When ADH is absent, the principal cells are impermeable to water at all. When ADH is present, depending on the concentration of ADH, the higher is this concentration, the higher is the opportunity to take water. Permeability of water increases. And permeability to water depends on water channels formed from aquaporins. Aquaporins are present in the principal cells inside, they are internalized. But under action of ADH, internalized aquaporins become inserted into the luminal membrane. Let's observe the process from the beginning. How the water channels finally make water to go into the cell. First, this side is luminal, where we need to take water by reabsorption, and this is the basolateral side where the membrane of cell faces blood, and ADH from blood can come and bind to receptors. And this is ADH molecule that binds to the receptor located in the principal cell. These are V2 receptors, vasopressin 2. And this receptor operates through GS protein and therefore activates adenylyl cyclase pathway. First, GS protein is activated after binding of ADH. Then adenylyl cyclase is activated and starts to produce cyclic AMP from ATP. Then cyclic AMP in turn activates protein kinase A. Protein kinase A phosphorylates the proteins. Perhaps the microtubules proteins are phosphorylated, and through this microtubules action, the aquaporins become transported from interior of cell to the membrane. And this is the type of aquaporin 2. And when aquaporins are inserted into membrane, they form water channels, and through these water channels, finally water may go inside the cell. Otherwise, membranes of the cells in the late distal tubule and collecting duct remain impermeable to water entirely. So there is no any water reabsorption if ADH is 0. When ADH is present, proportionally, permeability of water becomes higher and higher. Recirculation of urea. One important substance we haven't discussed yet, and this substance is low molecular substance, which is easily filtered. Let's start. It's fully filtered and remains in the filtered fluid. While fluid flows through the proximal tubule, it partially is reabsorbed, about 50%. But the remaining goes into the loop of Henle, and concentration of this urea becomes increased due to reabsorption of water in the descending part of the loop of Henle. But then it comes to the thick ascending portion where membrane is totally impermeable for urea. You see again, impermeable for urea is a big part of the nephron. Ascending thick portion of the loop of Henle, total distal tubule, and portion of the collecting duct even. And just lower portion, which passes through the inner medulla, is permeable for urea. So, due to gradual more high concentration reached in the late distal tubules and in the collecting duct, urea concentration becomes so high that it starts to go into interstitium down the gradient and becomes absorbed again into parts of the loop of Henle. It doesn't matter, both descending part and thin ascending part, both are permeable for urea, and urea easily enters. And so part of urea molecules constantly make rounds, make cycles. It comes from the loop of Henle to the distal tubule, then to the collecting duct, and from collecting duct it escapes into interstitium and again becomes included into the filtered fluid inside the tubule, inside the loop of Henle, and so it flows constantly. And due to this, urea is constantly present in the interstitium of the inner medulla, where it greatly contributes to the higher osmotic pressure, to hypertonicity. So part of urea is constantly reabsorbed, and it helps to maintain hypertonic interstitium, which contributes to water reabsorption in both descending part of loop of Henle and also in the water reabsorption in the lower part of the collecting duct, and part is excreted. So it's important that urea contributes to hyperosmolarity of the interstitium of the medulla, along with sodium, chlorine, and potassium reabsorbed from the thick ascending portion of the loop of Henle. Interstitium hypertonic due to sodium, potassium, chlorine, and urea. Secretion, last, the third process which we haven't described yet, and this secretion is the tubular secretion, release of substances from blood into the lumen of tubules. And it occurs in the proximal tubules, distal tubules, and collecting ducts. So you can notice that the only place where secretion does not occur is the loop of Henle. And mainly, under normal conditions, potassium and hydrogen ions are secreted, and their secretion helps to control normal, steady level of both potassium and hydrogen in blood, so that when potassium is higher, it's secreted in higher amount. When potassium becomes lower, it's less secreted, and similarly for H+ ions. So secretion of these ions is involved into maintaining the constancy of their concentration in blood. Again, how it occurs, we can recall from already considered processes in the principal cells, for example. Active sodium transport occurs by sodium-potassium ATPase in the basolateral membrane of the cells, while potassium is constantly brought into the cell and concentration is high. Therefore, potassium easily goes out into the lumen through channels, which is stimulated by aldosterone also. Principal cells of the late distal tubules and collecting ducts, mainly these processes occur. And you see potassium channels are present here, not only sodium channels, but also potassium channels, which allow potassium to go into the lumen. And so these ions are secreted, and secretion of potassium is also one of functions of aldosterone. And that's all about the general functioning of kidneys. The regulation is expected.